Section 1. The Golgi apparatus, a cellular processing hub. Within the bustling metropolis of a cell, proteins are constantly synthesized, modified and shipped to their designated locations. This intricate process of cellular logistics is overseen by a remarkable organelle known as the Golgi apparatus. Like a meticulous post office, the Golgi apparatus receives, processes, sorts and packages proteins and lipids. It ensures their timely delivery to various destinations within or outside the cell. The Golgi apparatus plays a critical role in maintaining cellular order and function. It acts as a central processing unit, ensuring that proteins are correctly folded, modified and labeled for their specific tasks. Without the Golgi, the cell would be thrown into disarray. Newly synthesized proteins would lack the necessary modifications for their function. Essential molecules would fail to reach their intended destinations. The Golgi apparatus is not a standalone entity. It collaborates closely with other cellular components. Its relationship with the endoplasmic reticulum, the site of protein synthesis, is particularly noteworthy. This dynamic duo works in concert to ensure the smooth flow of proteins from their production line to their final destinations. The Golgi apparatus receives newly synthesized proteins from the endoplasmic reticulum. It then embarks on a series of modifications and sorting processes. Understanding the Golgi apparatus is crucial to unraveling the complexities of cellular function. Its role in protein processing and transport underpins a wide range of cellular processes. Examples include cell signaling, membrane formation, and the secretion of hormones and enzymes. By delving into the intricacies of the Golgi's operations, we gain a deeper appreciation for the remarkable level of organization within even the simplest of cells. Section 2. Structure and location. The Golgi's strategic position. The Golgi apparatus, with its characteristic stacked structure, is strategically positioned within the cell's cytoplasm. It lies in close proximity to the nucleus, the cell's control center, and the endoplasmic reticulum, the protein synthesis machinery. This strategic location highlights the Golgi's pivotal role in coordinating cellular activities. The Golgi apparatus is composed of flattened, membrane-bound sacs, known as cisternae. These cisternae are arranged in stacks, resembling a pile of pancakes. Each stack, or Golgi stack, has a distinct CIS face and a trans face. The CIS face receives incoming transport vesicles from the endoplasmic reticulum, while the trans face buds off vesicles carrying processed proteins to their final destinations. This polarized organization of the Golgi stack is crucial for its function. It allows for the directional flow of proteins through the Golgi, ensuring that they undergo a sequential series of modifications. As proteins journey from the CIS face to the trans face, they encounter different enzymes residing in each cisterna. These enzymes act like specialized workers in a factory, each performing specific modifications to the passing proteins. The Golgi's close proximity to the nucleus and the endoplasmic reticulum underscores its interconnectedness with these crucial cellular components. This strategic location facilitates the efficient transfer of genetic information from the nucleus to the ribosomes on the endoplasmic reticulum, where protein synthesis takes place. It also ensures the rapid delivery of newly synthesized proteins from the endoplasmic reticulum to the Golgi for further processing and sorting. Section 3. From synthesis to sorting, the Golgi's processing pathway. The Golgi apparatus functions as a cellular post office. It receives, processes and sorts proteins synthesized in the endoplasmic reticulum. Think of the endoplasmic reticulum as a factory churning out raw materials. The Golgi apparatus then steps in as the processing and distribution center, ensuring each product is packaged and shipped to the correct destination. The journey of a protein through the Golgi apparatus begins with its arrival at the CIS face. Incoming transport vesicles from the endoplasmic reticulum fuse with the CIS Golgi network, delivering their cargo of newly synthesized proteins. Once inside the Golgi, proteins embark on an elaborate journey through the cisternae, encountering various enzymes responsible for their modification. As proteins traverse the Golgi stack, they undergo a series of post-translational modifications. These modifications, akin to adding stamps and addresses to a letter, are crucial for protein function and sorting. One of the key modifications is glycosylation, the attachment of sugar molecules. Glycosylation plays a vital role in protein folding, stability and recognition. Other modifications include phosphorylation, the addition of phosphate groups, and sulfation, the addition of sulfate groups. These modifications can alter protein activity, target them to specific cellular compartments, or prepare them for secretion. The Golgi apparatus acts as a quality control checkpoint, ensuring that only correctly modified proteins proceed to the next stage of the cellular journey. Section 4. Vesicular transport. The Golgi's shipping system. The Golgi apparatus employs an intricate system of vesicular transport to ship processed proteins to their final destinations. These tiny, membrane-bound sacs act as cellular vehicles, ferrying their precious cargo throughout the cell. The Golgi's shipping system ensures that proteins reach their intended targets, whether it's another organelle within the cell or the extracellular environment. Vesicles bud off from the transface of the Golgi, each carrying a specific set of proteins destined for a particular location. Imagine these vesicles as delivery trucks leaving the Golgi's sorting center, each loaded with packages addressed to different parts of the city. To ensure accurate delivery, each vesicle is coated with specific proteins that act as address labels. These protein coats, such as copii and clathrin, help to select the correct cargo for each vesicle and direct them to the appropriate destination. For instance, vesicles destined for the lysosome, the cell's recycling center, are coated with mannose 6-phosphate receptors. These receptors recognize specific sugar modifications on lysosomal enzymes, ensuring their proper delivery. Once a vesicle reaches its destination, it fuses with the target membrane, releasing its cargo. This fusion process is mediated by specific proteins called snares, which act like docking stations, ensuring that vesicles only fuse with the correct target membranes. The Golgi's shipping system is a marvel of cellular logistics, ensuring the timely and accurate delivery of essential molecules throughout the cell. Section 5. Golgi dysfunction. When the packaging plant malfunctions. The Golgi apparatus, despite its complexity, is remarkably efficient. However, like any well-oiled machine, it can malfunction, disrupting the delicate balance within the cell. When the Golgi's operations go awry, the consequences can be severe, leading to a range of diseases. One such disorder is congenital glycosylation deficiency, a group of inherited metabolic disorders caused by defects in the glycosylation process within the Golgi. These defects can lead to a wide range of symptoms, including developmental delays, seizures, and blood clotting problems. The malfunctioning Golgi apparatus disrupts the proper modification and sorting of proteins, hindering their ability to function correctly. Neurodegenerative disorders, such as Alzheimer's and Parkinson's diseases, have also been linked to Golgi dysfunction. In these conditions, the accumulation of misfolded proteins, often due to impaired protein degradation pathways, can overwhelm the Golgi's processing capacity. This backlog of misfolded proteins can disrupt the Golgi's structure and function, further exacerbating the disease process. The Golgi apparatus is also a target for certain pathogens. Viruses, for instance, can exploit the Golgi's machinery to replicate and spread within the cell. By hijacking the Golgi's vesicular transport system, viruses can disguise themselves in cellular membranes, evading the immune system and spreading to neighboring cells. Understanding how pathogens exploit the Golgi is crucial for developing effective antiviral and antibacterial therapies. Section 6. The Golgi's legacy, Camillo Golgi and beyond. The discovery of the Golgi apparatus in 1897 by Italian scientist Camillo Golgi marked a pivotal moment in cell biology. Using a staining technique he developed, Golgi observed a network of interconnected sacs and tubules near the nucleus of nerve cells. This groundbreaking observation, initially met with skepticism, laid the foundation for our understanding of this vital organelle. Golgi's staining technique, known as the black reaction, allowed him to visualize the Golgi apparatus with unprecedented clarity. This technique, which involved staining cells with silver nitrate, revealed the Golgi's intricate structure, distinguishing it from other cellular components. Golgi's meticulous observations and detailed descriptions paved the way for future generations of scientists to unravel the complexities of this cellular organelle. Today, the Golgi apparatus stands as a testament to the power of scientific inquiry and the importance of fundamental research. The legacy of Camillo Golgi lives on, not only in the organelle that bears his name, but also in the countless scientific advancements made possible by his groundbreaking discovery. The Golgi apparatus, once a mysterious cellular component, is now recognized as an essential organelle, crucial for maintaining cellular order and function. Its role in protein processing, sorting and transport underpins a wide range of cellular processes, impacting everything from cell signaling to membrane formation. As we delve deeper into the intricacies of the Golgi's operations, we gain a greater appreciation for the remarkable level of organization within even the simplest of cells.